Hi guys, Tracy again. Welcome to Parasitology lecture. We're a little bit non-traditional today in that unfortunately I'm still out of town, but I still want to cover the necessary information, as with our 15 weeks together we certainly have a lot of parasites to cover, so I don't want you to miss out because of my absence. So because of that, I'm going to do an online lecture today that Paula is going to give you access to this site. It's also a YouTube video. So if you need to reference it in the future, by all means, you have that resource available. I am very passionate about parasites. I think they're quite fascinating in that they're very resilient. They have never been on the endangered or extinct list, so that tells you how profound of an impact they can have on people, animals. Parasites can inhabit any part of a person or an animal's body, and we'll get to know a whole bunch of them throughout our semester together. Paula has gone through the textbooks. There is also a brand new textbook that I have included on the syllabus, but I have not put a picture here. We're going to utilize all of these textbooks throughout the semester in both lab and lecture. This lecture is very profound. There's a lot of information, and because of that, we're going to cover the main facets, the main philosophies, the main concepts of parasites in the lecture, but we also have some homework, some other assignments that are going to help the learning experience and learning these parasites. So because of that, you'll have to direct yourself. I'm going to help you direct yourself so that you know what to study, so that you can learn all of this information. To give you a little heads up, we're going to dedicate most of the semester to small animal parasites. And when I say small animals, I'm reflecting cats and dogs. But we are also going to discuss production medicine towards the end of the semester, so that you have a good basis going into large animal medicine, that class. But in reality, if you understand the concepts of a parasite and how it can affect a dog, you can understand how that same parasite can affect, let's say, a human. They might have a different name, a different scientific name, but nonetheless, they will act the same. So our dedication is to small animal medicine, a little bit towards the end of the semester, large animal medicine. But again, if you understand the concepts on how they affect small animals, you can apply that to any breed of animal, species of animal on the planet. I don't have my clicker with me, so I have to go back and forth to the computer. I put this picture, this PowerPoint is available, as are all of the other PowerPoints. They are available on eCompanion Doc Sharing, and you'll notice that they are there right now. And I'm going to update eCompanion as soon as I return to school and have access to it. So I'll put a copy of the syllabus just in case you happen to lose it or your dog eats it. So you have a great number of resources available to you on eCompanion. Parasites. We're spending 15 weeks on parasites, so they probably have a very profound effect on the veterinary community. And that's true. And in just looking at the Kitchen household, not including Gary, but we have Phoebe. If you don't know Phoebe, I'm sure you will, and likewise Floyd. I have two dogs who, ever since I acquired them from different rescues, I have made a point of trying to keep them free and clear of parasites. Because what you will learn today, you'll learn a lot of information from this lecture, but what's most important is that you understand what a parasite is. And I'll keep reviewing that. A parasite is something that can live inside or on top of the animal, like their hair and their skin, but offers no benefit. So it doesn't bring anything to the table. If anything, it takes something away from this animal. And I'm going to refer to this animal as the host. In the next couple of weeks, you will learn that puppies and kittens are pretty much born with some parasites because their mother has given it to them. So from the first day that I acquired all of my animals, and by the way, this is Jimmy James Jr., this is Junior, he is my hunting animal, which throughout the semester you'll learn that if you are an active hunter, and by hunting he is eating raw meat, those delicious bunny rabbits and birds and mice, delicious. By consuming that raw meat, it puts him at risk for a variety of other parasites. Special Ed, on the other hand, he doesn't go outside because he's special. He's kind of gimpy. But even so, Junior can bring in parasites. I can bring in parasites if I don't decontaminate myself after work. Parasites can enter my home. So even though he doesn't go outside, parasites can come in other ways. So I want to keep them protected. We are right now heading into mosquito season, and so mosquitoes can transmit a great variety of diseases, and we're going to spend a single module, 2 hours, on heartworm disease, which is a disease transmitted by the mosquito, and that's our state bird right here. So these are, again, more to come, 15 weeks of parasites, and certainly protecting our animals, not just our animals, but our clients' animals, keeping them safe from these harmful parasites. Again, they offer no benefit. Throughout the semester, we are going to recognize different parasites by their common name and by their scientific name. We are going to recognize parasites by different symptoms that they can cause in these animals. Again, our focus will be on cats and dogs. If you had a patient present with a given symptom, it might make you suspicious of this parasite. And to diagnose this parasite, we run particular tests. So we'll get an experience with running different tests, and you'll also do a variety of these tests in parasitology lab and also other classes like vet disease lab. The test is going to yield a proper, accurate diagnosis. And if you have an accurate diagnosis, the veterinarian will then understand what is necessary to correct that parasite, to kill that parasite, to eradicate that parasite, hence effective treatment. Versus if we didn't have an accurate diagnosis, the veterinarian, and you of course are playing an integral part of this, are taking guesses as to what medicines to choose. Once we have prescribed medicine to kill that parasite, that's the goal, because again, they offer no benefit. If anything, they are going to be harmful to the animal. We want to make sure that our treatment has been effective in doing so, killing that parasite, so we always recheck. So throughout the course of the semester, this is going to be our flow chart, recognizing symptoms to go through this process of yielding an effective diagnosis to put the animal on the appropriate therapy. And we're going to learn about different drugs in this class that kill parasites. So what is a parasite? As I mentioned, a parasite is something that can live on the host. We would refer to those as external parasites or ectoparasites, or it can live in the host. We would call those internal parasites or endoparasites. And again, they don't offer any benefit. They are utilizing energy or something from the animal. Again, they're taking something from that animal, that host. That host is a term in which that parasite is residing on or in. Parasite doesn't contribute to the survival of the host. We do not need them. If anything, they cause us harm. Now, sometimes you might be harboring a parasite. The Centers for Disease Control, the CDC, it's a wonderful resource. I would encourage you to use it in this class and in other classes, microbiology, vet disease, SAM2, etc. The CDC says that 50% of humans have roundworms. And this is going to be our first parasite that we discuss next week because it's very common. And a roundworm is a worm that is round in diameter. It looks like a piece of spaghetti. So 50% of you right now in this classroom have roundworms. And where they live as adults are in your intestines. Now, I haven't seen you yet, but I can only assume that you look healthy. So why aren't you exhibiting any symptoms of that roundworm? Because again, 50% of you have roundworms and just don't know it. And it's probably because you're eating enough to feed you and your parasitic worms. And hence, because of that, everybody's happy and you're not seeing any symptoms. But let's say you lived in an underdeveloped country who struggles with poor nutrition. That same individual that had roundworms, that parasite is taking energy from you, sucking nutrition from you, stealing nutrition from you. And because of that, you're probably more likely to struggle with side effects or symptoms of that parasite. Kind of neat. We'll talk about how parasites are so successful at being parasites. Again, as I mentioned, they're never on the endangered list. They're never on the extinct list like many other animals and plants. So how do they become so successful? Well, ultimately, two main reasons is they produce a large number of babies. I put babies in quotation because we don't call them babies. We might call them an egg. We might call them something else, an oocyst, a trovazoite, something. But if we have mom and dad that get together, or an organism that is capable of reproducing, they're going to reproduce a large amount of offspring, knowing that most of those offspring might not survive, but some of them will. And again, different elements like weather, temperature are going to contribute to their successfulness. Many of the parasites have characteristics that are going to help them be successful. This is a classic roundworm egg, and many of you have already had lab or will have lab, and you will be introduced to this parasite module 1. This is a classic roundworm egg, more specifically a toxocar egg. And what roundworm eggs all share in common is that they have this very thick outer membrane. And this is the actual parasite inside here. So this parasite, this baby, has a very large protective outer winter coat. So what a wonderful characteristic, especially going into cooler climates. We do have that nice winter coat, so they're not bothered by the cool weather. Cold, 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 probably that jacket isn't warm enough. But certainly many of these parasites, because of these characteristics, can withstand some harsh elements. Unfavorable conditions, as it says. Many of these parasites, not all of them, obviously we're going to talk about ways to protect yourself and your clients from the possibility of zoonoses. Of course, many of these parasites, the first part of our class are going to be dedicated to gastrointestinal parasites. So somewhere between food in and poop out, the worms or these organisms are going to reside somewhere in that cavity. So to protect ourselves, knowing that these parasites reside in the gastrointestinal system, the waste product of the gastrointestinal system is feces, we're going to detect a lot of these parasites in the feces with a fecal examination. And so when you handle feces, you certainly want to practice universal precaution. So anytime you are handling feces, you're going to be wearing gloves. Even because you're wearing gloves, you're going to wash your hands frequently. So even after when you take off those gloves, you're going to wash your hands. In lab, if you don't wash your hands after leaving the laboratory area, your daily points will be affected. This is serious. Now, many of these zoonotic diseases, they're not going to kill you, but they certainly are going to cause you harm or some sometimes devastating side effects or symptoms. All samples, again, universal precautions, because we're going to consider all of them to be zoonotic, we are going to wear gloves. Not all of them are going to be zoonotic. For the most part, the majority are not. But you don't know that just looking at a fecal sample. At any time, don't put your fingers in your mouth. Don't touch people. They go, oh my God, look at what I found on the microscope. So please don't touch yourself or anyone else. And again, after you remove your gloves, you're going to wash your hands. As I mentioned, we are going to detect these gastrointestinal parasites via the aid of a fecal examination. Now, looking at the example that I just provided in that roundworms, our first category of parasites we're going to tackle next week. This is a classic roundworm adult. And this roundworm adult lives in the intestines. It actually specifically lives in the small intestine. And if you've taken comparative anatomy, and don't worry if you haven't, you will be covering that this semester if you are in comparative anatomy. The small intestine is where you have a large, significant amount of absorption of nutrients. Specifically in your small intestine, where guess where these guys live? They live in that area. So as you take in nutrition, your body breaks down that food, and in that small intestine it's ready to be absorbed by your body. Well, here they are stealing that nutrition. It's very rare, uncommon. I shouldn't say rare. It's uncommon for you to find these adults in the fecal examination. We'll talk about reasons why you might see adults later on in a few slides. But we are not going to rely on finding these guys, the adults, in our fecal examination. Instead, we are going to find their babies. This is a roundworm egg. And with that, again, we're going to do 3 distinct tests in our fecal examination in lab. But I thought that I would also review it in lecture as well. We have parasites that live in the gastrointestinal system. Yes, the vast majority of the worms and other parasites that we're going to discuss reside in the GI system. But we have other parasites that live in the biliary system. Biliary is something that produces bile, and in anatomy you will know or know that the biliary system is referring to the liver. The liver fluke, the liver fluke is probably the most common one I can think of. Respiratory system, so somewhere in the lungs. How about the lung fluke, the lung worm? There's a couple there. We have parasites that reside also in the urinary system. Capillaria aerophilia is the scientific name of the common bladder worm. But for the most part, our focus are going to be on the gastrointestinal parasites, at least for the first part of the semester, the endoparasites. We might find in the fecal examination things that look like parasites but are truly not, and especially in spring semester, when things are pollinating out there, we are going to see a lot of pollens that are infiltrating our fecal sample. So that makes it a little bit more challenging for you, but in lab we'll talk about ways, characteristics of a parasite that will help you say this is a parasite and apply it to something else and say, well, it doesn't share those characteristics, so it mustn't be a parasite. We can find adult worms. Now, a few seconds ago I said it's very uncommon for you to find adult worms, but especially with those round worms, those pieces of spaghetti, again, they like to live, they prefer to live up in the small intestine. So what can be some reasons of why they might be expelled? Because believe it or not, when those worms leave the small intestine, they are not in their home, and hence not getting the nutrition that they need. And if you don't get the nutrition that you need, you will die. So what could cause them to leave the gastrointestinal system? Well, a case of enteritis. So what if the dog or the cat has diarrhea and all the spasmings of that tract forces them out? That's not good for them. They will die. Another reason why you might find adult worms is you've diagnosed a parasite, and when you diagnose a parasite, what is the next step? You kill the parasite. So anytime you put an animal on some parasiticide, something that kills a parasite, that parasite could evacuate and leave. And again, you could see that in the fecal examination. You can find spurious parasites. We're not really going to focus on spurious parasite, real parasites, but they're in the wrong host. For example, Toxocara canis, common parasite you will learn about in lab number 1. Toxocara canis is the roundworm of dogs. What if you found Toxocara canis in a cat? Well, it's a real parasite, but it's supposed to be in a dog. Again, that's all I want to say. It's no big, leave those to the parasitologist. In your fecal examination, your ideal sample should be fresh. And so right now we've got nice cool weather, but some of the evenings are still cold. Possibility of freezing? Probably not. But you don't want them to freeze. You don't want it to get too hot. You want to keep it nice and cool. To do that, if you do ask an owner to bring in a fecal sample, ask them to put it in a tightly sealed container so again it doesn't spill or the odor doesn't get too profound for them, and you want to have them keep it in the refrigerator. What can happen if it's an old sample? They can start to desiccate, so dry up, and because of that it makes it hard to perform the test, but more importantly parasites can leave the sample or simply die. We can also have parasites, like let's say you have a fecal sample out on the grass outside. Parasites can enter that sample. That doesn't mean that the host is affected, it just means that they've infiltrated the sample after the fact. So ways to collect a fecal sample, you could have the owner collect, which doesn't mean they're going to stick their hand in the dog's anus or the cat's anus. They're going to allow the animal to defecate in the litter box or outside, wherever they do their business, and then they collect the sample, put it in a container of some sort. As technicians, we can remove right from the rectum, and so you might do that with a gloved hand, put a little bit of lubricant on, and collect your sample. You can also perform an enema. An enema is an infusion of a liquid solution through the anus, so you're going to have it become very diluted, but nonetheless, you can still perform the test if the doctor asks for that test. We can store that sample in the refrigerator for a couple of days. Again, the host, think of your intestines right now, nice and warm, 100 to 102.5 for animals, degrees Fahrenheit. And so in the refrigerator, that's a lot cooler than their natural environment. So after a few days, they will start to die. If you wanted to preserve it indefinitely, you're going to use a 10% formalin, a buffered phosphate formalin. And that's for preservation long term. Again, as I mentioned, you're going to transport that sample in a leak-proof container so it doesn't spill in your car, that you identify it. And here at Argosy, you're going to put the patient's name and the date of collection. That's going to be helpful to see how old the specimen is, because you now know what happens with age. And again, you want to prevent it from getting too odorophilous. You can certainly watch this fun little YouTube. It's from, a television show, Scrubs. It's really cute. Having said that, let's go back to our fecal examination. We have 3 distinct tests for the fecal exam. And this is something you're going to tackle in the subsequent weeks in lab. 3 distinct tests. So you have taken veterinary laboratory procedures and reflect back on a complete urinalysis. 3 distinct tests. You're going to see some similarities between a urinalysis and a fecal exam, but there are some differences, obviously. Gross physical exam. So with your urinalysis, you looked at the color, you looked at the clarity, you appreciated the odor, and you also did a specific gravity. With a fecal exam, your gross examination is going to look at the color of the feces, assess the odor of the feces, and also the consistency of the feces. Again, I'll go into the slides a little bit more depth later on. Direct smear is another distinct test that is a microscopic test, as is the flotation. And I actually have shown you here a common flotation device called the fecalizer. Again, the gross examination, what does it do? It looks at the color, the consistency of the feces, you assess the odor, and that just kind of gives you an overview, information of the condition of the animal's GI system. So think of the last time that you went into the bathroom, a number 2, and it was really putrid. That told you that you ate something that maybe didn't agree with you. Or if you had diarrhea, that loose feces, it tells you that you didn't eat something that it didn't agree with you. So again, a very vague, very subjective, general view of the health of the GI system. By no means are you going to look at the color, appreciate the consistency and the odor and say, this animal must have some sort of colon cancer. Absolutely not. Just very vague, very general. So we need to report those things as I mentioned. So if this were my sample, I certainly can indicate the color here. Now brown, there's so many shades of brown, and you can see here that we've got different shades of brown. We've got some light brown and we have some dark brown. We try not to use food analogies. I know I'm classic at talking about food analogies, like this looks more like dark chocolate, this looks more like milk chocolate, but I'm never, ever, ever going to record that in the animal's fecal examination. We're going to use professional laboratory terms. Consistency, I'm going to say that this is formed. I can't assess the odor right now, but you would obviously in a real life setting. Now, this miscellaneous, let's talk about this a little bit more in just a few moments. Color, here's some really great helpful indicators. Brown is normal. Again, you can be more specific, light brown, dark brown. Here are some colors that would reflect some abnormalities. So for example, what if you had a very light brown, a very tan colored fecal exam? That would give you some insight that possibly that animal is consuming some milk products, some dairy products. Again, at this time, it gives me an excellent opportunity to ask the owner, what is this your pet being fed? And ask them, is there anything other than their classic dog food that they eat? Well, in the morning they get some of my leftover cereal milk. Even though it doesn't seem to be affecting the health of this fecal sample, again, if it did, that would be an excellent opportunity for you to educate the owner that animals, after they're weaned from their mothers, are pretty much lactose intolerant. They shouldn't be fed dairy products. So a wonderful opportunity to educate the owners. You're not going to degrade them and say, hey, you stupid idiot, you can't feed dairy products to animals. They're lactose intolerant. No, you're going to do it in a professional manner. I want to spend some more time on red. Now, we have light red and we have dark red. Now, not light red, bright red. Bright red like an apple, like a fire engine. Dark red, think of burgundy. I'm trying to find some reds in this classroom. Those, either way, that red tells you that there's bleeding somewhere in the gastrointestinal system. If there was a bright red blood found in the feces, that tells you that the bleeding occurs more towards the anus, because that's where the feces comes out. So it's fresh blood. Anus, colon bleeding, versus that dark brown to black, that burgundy, that dark blood. That tells you that the bleeding is more proximal, more stomach, more small intestine. So again, if you ate something sharp, Floyd just ate a bone, unfortunately, and thankfully it was able to pass, but it must have cut his rectum or his anus a little bit because there was some bright red blood in it. And he was certainly a little bit uncomfortable in passing that bone. Versus bleeding upper, it's going to look very, very, very dark and tar-like. Consistency, again, this PowerPoint is, I wanted to provide you some helpful terms that you can utilize in lab. You can't say unprofessional terms. So you can't say, oh, it looks like loose crap. No, you can't say that at all. You wouldn't want that. You want a professional. You want your technicians to be professional. Whoever's taking care of your animals, you want them to be professional. Again, odor is going to vary depending on the animal. So our focus is going to be small animals, but towards the end of the semester, we're going to do production animals like horses and cows. And horse feces has a very distinct, different smell compared to that of dogs and cats. And it makes sense because of their nutritional requirements. Horses and cows, they consume plant material. They're herbivores. Versus cats and dogs who are carnivores. They're predominantly eating proteinaceous things. Humans, a lot of variety in our diet, and we consume a lot of proteinaceous things as well. So in theory, a horse fecal sample should smell like plants, should smell like grass. Versus dogs and cats, you're not going to get that odor appreciated. So my point is, throughout the semester, we'll learn what is normal and what is abnormal, because that's a huge purpose of being a technician for that part. So gross examination, color, odor, consistency, and the fourth thing that we are going to assess is anything miscellaneous. And so this is where you have to dig in into your fecal sample, because sometimes things are not overtly on the surface of the feces. So you might grab some applicator sticks and dig in. Here are some things that you might find. In the fecal sample that I just mentioned with Floyd, I found some bone particulates. So once I identify this miscellaneous item, I also want to quantify it. So you can use a grading scale like small, medium, moderate, large, or you can use a grading scale that the AVMA, American Veterinary Medical Association, wants you to use. 1+ through 4+. 1+ means that there's a small amount in it. 4+ means that there's more of that in it than there is feces. And again, you can gauge in between. But here's some, if you saw mucus. Mucus, I hate to use the word, but it looks like snot. And that tells you that there's colon irritation. So a lot of these, in Floyd's fecal sample, I also saw some mucus. Something was irritating his colon, and I was able to detect what was irritating it because we had some bones. So anything miscellaneous that you find, you also want to quantify. Phoebe's feces not too long ago, some birds go into the bird feeder and they spill a whole bunch of bird seed onto the ground below. Well, Phoebe goes and eats it. So guess what? She probably had 2 to 3+ bird seed. What a perfect opportunity for you to educate that client. Your dog eats bird seed. Let's try and prevent that. The second test in the complete fecal exam is the direct smear. Direct smear is a simple test that you'll learn in lab. You take a clean glass slide, you label it with the necessary information, and you put a very tiny, tiny amount of feces on it, and you dilute it with water. Very, very dilute, and then you put a clean glass cover slip. You're going to observe your cover slip under 40 power, and you're going to quantify the amount of bacteria that you see. You're going to quantify it as what is your percentage of round bacteria to rod bacteria. And you're also going to look for this parasite called Giardia, because it's best identified on 40 power with the aid of a direct smear. The fecal float is the third test. There's a variety of flotation devices. Right over here, you are seeing the device, the flotation device called the fecalizer. When you do your reading assignments, you're going to see pictures of, like, say, the ovitector. In lab, we're also going to be accomplishing the centrifugation flotation, using the centrifuge to perform a flotation, which is actually the best out of all of these. But the purpose of it is using this fecalizer. This is the test device that we're going to utilize for the first couple of weeks. We're going to take out this center piece, and it has little holes. It looks like a colander. We're going to stick some feces on the bottom. We're going to mix it with a flotation solution, so not water. This is a substance that is usually sugar-based, salt-based. There's a lot of commercial flotation solutions available. And the purpose of it, if it's within a specific range of specific gravity, if there are parasites in the fecal sample that are going to be less dense than the flotation solution, they will rise to the surface, float to the surface. And so we're going to take feces, we're going to mix it with flotation solution, we're going to stick this device back in, and through those holes, only small little things, so the big chunks like of litter or plant material or whatever is going to be stuck to the bottom, unable to make it through those little holes. They rise to the surface because they're less dense, and we are going to have a nice little cover slip waiting on top to have those eggs collect on the cover slip. Again, you'll learn more about it in lab. And again, it's a good test. This is a good test for the detection of parasites. Even better is going to be the centrifugation. Oopsies. So, 15 weeks, we're going to discuss parasites, obviously. We're going to discuss parasites by their common names and by their scientific names. We are going to learn a lot about how parasites can affect small animals, and because of that, we can take that information and apply it to any animal that you might happen to deal with. And we have the luxury of working with a great variety of animals, being veterinary technicians. How can parasites affect hosts? Well, as I mentioned, for example, just using the example of the roundworm, this roundworm resides in the small intestine, is going to steal your food. We're going to talk about tapeworms in a couple of weeks. They also reside in the small intestine, and they are really good at sucking the nutrients so that you don't get them. They like to steal and really have a strong competition for this food. Can you imagine being a young dog infected with roundworms? Do you see how it could affect their growth if this parasite went undetected? Some parasites are bloodsuckers. Some, not all of them. This is an example of a hookworm. It's actually related to the roundworms that you just saw, kind of a cousin, let's say. And a hookworm, this is a picture very zoomed in of their face area. And it looks like he's got teeth. They're not teeth, but they're anchoring mechanisms. And so this particular worm also resides in the small intestine, is there to suck any nutrients that you might take in through your digestive system. But he or she also needs blood for his survival. And so when he needs a blood meal, she needs a blood meal, they're going to latch onto the small intestine and suck some blood. Not all parasites are capable of doing that. You know others, flies, mosquitoes, ticks. If you've lived in the upper Midwest, you're probably quite familiar with ticks. Fleas are very common here in the Midwest. So not all parasites suck blood, but some of them do. And again, if I had hookworms, the CDC says 30% of people are infected with hookworms. If I had a couple worms living in my guts, they are capable of sucking like 1/10 of a mil of blood every day. Is that going to make me anemic? Probably not. But what if that same young puppy or that small little kitten had a couple hookworms? Could we be anemic? Absolutely. Anemia, a lack of red blood cells. We have some parasites, when there are so many of them, they can obstruct the area in which they reside. So here's a classic example of the organ, the muscle organ in your chest that is responsible for pumping blood, to distribute all that blood and all the nutrients to the cells of the body, like oxygen, etc. Now, if this heart is infiltrated with worms, what do you think do you think it can pump effectively? Absolutely not. This animal will die of congestive heart failure. There's a lot of worms there. Whew. Any foreigner to the body, any foreigner, so not just parasites, but bacteria, viruses, fungus, if they enter a host, the immune system is going to, if working properly, is going to activate and say, foreigner has gained access. When we have a parasite entering a host, the immune system is going to recognize that, activate, and they're going to send out specific white blood cells to try and kill that parasite. And this white blood cell that is responsible, that we're going to see more commonly with parasitic infections, is the eosinophil. Many of you are in hematology or will be in hematology, and you'll learn about the different white blood cells. But this eosinophil, when you have a patient who has a parasitic infection, you're going to see a higher number of eosinophils, eosinophilia. Another reason you can see eosinophilia is because of allergies. So if you do a complete blood count on an animal, a blood test, looking at the red blood cells, the white blood cells, and platelets, I don't see any platelets on this smear, you do a CBC to count those and you see eosinophilia, you then know, do we have a parasite problem or do we have an allergy problem? We have some parasites that animals might be allergic to. This is a very common problem at the University of Minnesota in their dermatology specialty area. This is probably the most common thing that they see, and this is called flea bite allergic dermatitis. And this dog had an allergic reaction to the flea bite, more specifically the flea saliva. And because of that, he scratched himself raw. This is actually healed pretty well. So you can have any animal that could be allergic to any parasite. But again, this is the most common one. We can have parasites that cause external damage. We've just talked about some internal damage, but externally, think of your flies, your mosquitoes. Here's a cow, And when you are in the luxury of your own home, looking at the PowerPoints on your computer, you'll be able to see that this bovine here, this cow, has a lot of nodules. And if we were to take up the hide, we could see all of these larval stages of the fly burrowing underneath. So if I was a farmer and all I bred were cows, to sell their hide and other products, you can imagine I'm not going to be making a lot of money to sell this for nice shoes or nice purses. You know, Coach Purse is not going to buy this. I probably would buy it, but they probably wouldn't. We can have parasites that can transmit a lot of other organisms. So for example, the tick here. The tick, if you have paid attention to the news, this year, last year, whatever, you hear about a lot of tick-borne infections. And the tick picks up an organism while he or she sucks blood from that animal, picks up an organism, and then when he's hungry again and sucks blood off of another animal, he transmits that organism. Mosquitoes, you've probably heard in the news West Nile. West Nile is a mosquito-borne. The mosquito is transmitting it. So we'll talk about parasites who can transmit infection. Where do we find them? Well, and hence, how can they infect us or animals? We find parasites in the soil. That could be a very common area. So think of that dog defecating. He doesn't defecate in the toilet. He defecates outside. And what if you are playing outside and happen to accidentally touch that fecal matter? Now, it's not like this kid is going to put her fingers in the feces. What if the rain has washed away all the big chunks of feces and now the organism, microscopic organism, is in her sandbox or on the grass? You know, we're constantly touching these surfaces, and let's say you bite your nails like Tracy, you can infect yourself that way. Now, we are blessed with a wonderful water system, so you assume that the water that you are drinking is free of organisms, parasitic organisms, and so forth. But what if your water wasn't being filtered correctly? Then many of these parasites love water. You'll learn that Coccidia, Giardia, they love moist environments. And so you would just hope that your water is being filtered, and so it's safe to drink. We have some parasites that are found in your food sources. For example, there's a beef tapeworm, there is a pork tapeworm, there is a chicken tapeworm, there is a fish tapeworm. And so if you consume any of those products, and if they were infected with the infective stage, which is a microscopic stage, you can't see it. If that infective stage was infiltrating that meat product that you were consuming, and you didn't cook it medium or above, medium well will kill the parasite. So if you like your sushi, or if you like your steaks medium rare, then if that meat is contaminated, you could be ingesting it. Again, that's also other animals. If you are hunting, like for example, Junior, my cat, who's a hunter, puts him at risk. So if that meat that he is consuming is infected with the infective stage, he could be consuming it. How do parasites gain entry? Well, we can ingest them. So just like I mentioned with the previous slide, the kid playing in her playground, play box, sandbox, whatever you call it, and getting your hands in the soil, on the grass, and then ingesting it. Water source, ingesting that glass of water, and if it's infiltrated with an organism. We have some parasites that can migrate through the skin. Not all of them, just a few of them, but some of them can have skin penetration. We have some parasites that can go through the placenta, and so in utero, mother can transmit the organism, the parasite, to her young. Another ways mom can share their parasites is through the milk, and that's transmammary transmission. Mosquitoes, West Nile disease, heartworm disease is a blood-borne parasite. So the mosquito, while ingesting a blood meal, distributes or transfers the organism. And then again, we have indirect transmission where the animal is consuming something else that is infected. And again, through the course of today, I'll share you what indirect is. How do we control parasites? So ultimately, strict sanitation. Now let's say this were a test and I said, what's one way to control parasites? And you said sanitation. That's not good enough. I need more specific. So by cleaning up environments with a proper disinfectant to, again, disinfect that area completely. That's more specific. Heat sterilization, like the autoclave. If you put the world in an autoclave, it's going to kill all organisms. Well, that's unlikely. We're not going to be able to put the world in an autoclave. But heat sterilization, when you get into surgery anesthesia, if you're doing surgery on a patient who has a parasite, and so all of the instruments that Dr. Hulse uses is then contaminated, you need to autoclave them. You need to autoclave them anyways, but that will make you confident that the parasite is dead. Refrigeration, as I mentioned, so if you are asking an owner to bring in a sample, a fecal sample, you can put it in the refrigerator for a couple of days. It's going to control the population of parasites. So, destruction of host or intermediate host. So let's say Toxocara canis, the dog roundworm. If we killed all of the dogs on the planet, the dog roundworm wouldn't be able to survive. It's a bad example, but it's a good example. Also, let's talk about heartworm. I just mentioned that heartworm was because of the mosquitoes transmitting it to the dog or to the cat. So if we killed all of the mosquitoes on the planet, there would be no heartworm. So just some examples. Nutrition in small animals and large animals is more of a preventative measure. So think of my dogs, Phoebe and Floyd. They're always getting into trouble. But nonetheless, if we can feel confident that they are on a full balanced meal, getting all of their necessary nutrition, they're less likely to scavenge and find food from other sources. And education. That's the purpose of the vet tech, is to educate clients on ways to control their pets from getting any sort of parasitic infection. Now, parasites can be broken down into two categories. The two categories are called obligatory parasites and facultative parasites. If you're taking microbiology right now, you're going to learn about facultative organisms, an obligatory organism, or facultative bacteria and obligatory bacteria. Obligatory is a term that means needs to live. And so when you have obligatory parasites, you're referring to an organism that needs to live as a parasite. And so let's just use an example of the dog roundworm, Toxocara canis. That worm needs to live in the dog. If there were no dogs present, what do you think is going to happen to the dog roundworm? It's not going to be able to survive. So without the dog, this roundworm is not successful. It's actually deceased. So needs versus facultative, which means it doesn't need to live off of that animal. So an example could be this fly. Flies suck blood for their own egg production and their nutrients. So they can suck blood, but if there's no blood around, they can find nutrition from other sources. So they don't need to live on or in another host at the expense of that host. An incidental parasite, some people call it an accidental parasite. It's found in an animal other than the natural host, but just, Toxocara canis, what if you found it in a cat? Sometimes it can develop the same way, but nonetheless it's still found in the wrong host. Again, you're not going to be tested on incidental, but you will be tested on temporary parasites. These are parasites that just stop, take something from you, and leave. So ticks, flies, mosquitoes. I've had boyfriends like this long ago, just kidding, that just take something from you and then just leave. It's a joke. Endoparasite, as I mentioned before, they live on the inside. So here's a classic example of intestines with these cylindrical worms inside. They're most likely round worms, most likely obstructed as well. Ectoparasites, they live on the outside, or these are the external parasites. Here's an example of a louse. This is more specifically Felicola, which is the cat biting louse. And you'll learn how to differentiate all of these parasites throughout the course of our semester. Permanent parasites are parasites that need to spend their entire life cycle on that host or in that host. And so let's go back to the roundworm. The roundworm, as adults, reside in the small intestine, as adults. If there's a female roundworm and a male roundworm, they're going to mate, copulate, and they're going to produce babies. The babies are expelled or excreted in the feces. Because this parasite leaves the host, it's excreted out into the outside world. It is not a permanent parasite. But many parasites are. The louse that you just saw is a permanent parasite. Every single stage needs to be on that host. All mites, this is a mite called Demodex. This, isn't it cute? It's cute, yes, but you don't want it. Well, actually, we'll get into that another time. But all mites, as adults, if there's a male and a female, these mites are going to produce eggs, which are going to mature into nymphs, or larvae, then nymphs, then adults. Every single stage is on that animal, which makes them a permanent parasite. Now, we have a variety of hosts. You're going to see that when I describe these, they all are very similar. So after I go through them, I'll give you some examples. Oh, darn it. I love these visuals, and I'm sorry that I got cut off here, but I'll walk you through it. And if you have the Forite book, the green book with the spiral bound, I love that book for a variety of reasons, in that they have all of these visual life cycles. And I'll talk about life cycles in a little bit. But if we look at just this visual, looks like we have an adult, like a human here, and it's pointing to the intestines where you see some worms. And so it's saying that if there is a male and female worm in here, the worms will be able to mate. And we are going to expel, it says here, feces, and we have the egg. So this person is pooping outside. I don't know, but just for fun, he's pooping outside on the ground. And in that feces, if mom and dad are present, we will excrete eggs in the fecal matter. You're going to see this egg is just going to mature a little bit. So when you see the picture, the egg is just maturing, continues to mature, and if another human accidentally ingested this egg or eggs, it could have the opportunity, if there's a male and female, or it could mature into an adult in the small intestine, and if there were a male and female, they could again continue the cycle and make eggs. So, going back to this definition of host, this is the primary host for this parasite. Primary hosts mean that it's the animal, the person, the animal, the breed of dog, the cat, the dog, the horse, the monkey, whatever, that supports the sexually mature form of the parasite. So when we talk sexually mature, we're usually referencing the adult stage. So where are the adults living? Specifically, the animal, not specifically the organ. So who is harboring those worms? And in this case, the human is the primary host, because we are able to support those worms. Now, nowhere in the previous life cycle, the one where the human, nowhere in this life cycle did you have any other animal. It was human, pooped out the eggs, and then ingested the eggs. No other animal thing was in that cycle. So let's look at this one now. Here we have the cat, and I don't know if you can see there's these worms in his belly. It looks like Junior. As we look at this life cycle, he poops out these eggs, and this egg is eaten by a rodent. It's located in the cycle. That tells me he is necessary for the success of this worm. This worm, this parasitic worm, needs this animal to complete its life cycle. Oh, isn't that amazing? This worm is defecated out. The eggs are then consumed by a rodent, like a mouse or a rat. And this cat eats this mouse. When that happens, if this mouse is infected with that, this cat will develop worms in his guts. That's cool. So, who supports the sexually mature form of the parasite? Who has the adult stage? The worms are in the cat. So in this example, the cat is the primary host. But this mouse is referred to as the secondary host, or intermediate, whatever you'd like to call it, because he is necessary to this parasite's life cycle. He is necessary and he's supporting the asexual form. So this little stage right here can't have sex. It's not a worm. And so once this egg infiltrates this mouse, there's an asexual form of the parasite in this mouse. It is then consumed. This mouse will never, ever, ever, ever develop worms. This worm, no. But once this cat ingests the asexual stage that's present in this mouse, we can then, only then, the parasite will develop into adult stage. Pretty cool, huh? So if you do not have a hunting cat, you don't need to worry about this parasite, this particular tapeworm. Special Ed, my cat with the gimp, he does not hunt. So therefore, I never have to worry about tapeworms. But Junior, on the other hand, he hunts every day. And so this is something that I have to treat him for, because he gets infected quite frequently. Now here are some other terms. Again, you're going to see a lot of similarity. A paratinic is known as a transfer host. And the key here is it just transfers the parasite, but it's not necessary. So looking at this diagram here, and I'm sorry that I got cut off. Here's a dog, and it's saying inside that dog there are worms. What kind of host are we? We must be a primary host. The dog has the worms, we're defecating out the egg. And let's follow this red cycle, this main life cycle. The eggs are defecated out, and if the dog accidentally walks in the grass and then licks his paws and then consumes that egg, that then can develop into a worm, and the cycle continues. But looking at these other things, they're not in the main life cycle. What does that mean? So let me explain. Here's the dog. We have the worms. We defecate out the eggs, but let's say it's consumed by a rabbit. This rabbit here would be known as a paratinic host. Inside the rabbit, the egg is still going to be able to survive for a period of time. However long that is, months to years, depending on the environmental conditions, depending on the health of the rabbit, whatever. It stays viable in this animal, but by no means does it ever grow up to be an adult. Like the worms will never be in this rabbit. It stays viable. It survives. And the ultimate goal for this parasite is to be consumed by a dog. This is an example of Toxocara canis, the dog roundworm. So this is a parasite that is not necessary to the survival. The parasite was accidentally ingested by a rabbit. The parasite's like, dang it, I didn't want to go into a rabbit. I wanted to go into a dog. I hope that dog eats that rabbit. And if the dog eats the rabbit, the tissue that contains this stage, this parasitic stage, then shabam, now, only in the dog will it be able to grow up to be an adult worm. And if there is a male and female worm, the cycle can continue. Let me explain this third one while we're here. Dog has the worms, defecates out the worms. What if a human consumed it? So let's say you were out gardening and you didn't have gloves on, and then you bit your nails or did something and accidentally consumed this parasite. again, this parasite is never going to grow up to be a worm in you if you consumed it. You're going to have this this stage that stays in particular tissues in your body. Yeah. It can cause you harm. It can cause you actually eye problems. It can cause you skin problems. But it is never going to grow up to be a worm. This is an example of a zoonotic infection. So this rabbit here is an example of a parotenic host or a transfer host. He aids in the infection, but he's not necessary. Like that mouse was absolutely necessary to that tapeworm example. A vector. When you, we have biological vectors, we've got mechanical vectors. We're going to pay more attention to the biological ones, in that when we're talking about biological vectors, we're usually referencing something that transfers the disease from one animal to the next. And a vector, for the most part, 90+% of the time, is going to be talking about an arthropod. So some sort of bug, a mosquito, a fly, a tick, a spider. They picked up the organism from somewhere and they're just transferring it to you. So this is another example of a transfer host, but when it's a bug like this, we would instead of using parotenic, we would use vector. All right, I'm going to blow your mind. So we have primary hosts, we have intermediate hosts or secondary hosts. We've got transfer hosts that can, if they are an animal, just a general animal, we might call them a parotenic host. If it's a bug, like a spider or a tick, we might call it a vector. But if this transfer host has a spine, and is responsible for transmitting organisms to other animals and including people, we might reference them as a reservoir host. And so, for example, the beaver is a common reservoir host for Giardia. He transfers the organism. So you're going to see me say the word transfer a lot, but depending on the type of animal that transmits it, is going to help guide you. Do I use parotenic, do I use vector, or do I use reservoir? Now, we've got two basic types of life cycles. Now, you've seen, again, it's cut off, it's annoying me, but we have two basic types of life cycles for all parasites. Direct and indirect. Direct means that there's only one animal. Let me explain. With this visual, you can see here that you have a horse, and it looks like in the stomach there are some worms. We defecate, if there's a male worm and a female worm, we defecate out the parasite, and if ingested by another horse, then there's no other animal in this life cycle. There's none. That's always a direct life cycle. Now, if at any time there's another animal in that life cycle, it's always indirect. So, you can see here with this life cycle, there's a person and there's a cow. Because there's two different animals, that would be four, two different animals, or more than one. I should explain, more than one, because sometimes you can see 3 or 4 different creatures in this life cycle. It's always indirect. But let's go. Oh, I think someone's here. I'm going to put it on pause. Oh, they're gone. Because, let's use our knowledge from the previous hosts. So because there are two animals, it's an indirect life cycle. Now, who is the primary host and who is the secondary host or the intermediate host? Who supports the adults? Well, it looks here that here's the adult stage. It looks like a flat segmented worm. So if you've taken lab, I want you to guess what sort of worm this is, a flat segmented worm. And this worm is in this person. So that makes this person a primary host. The person defecates out the parasite. Oh, they're pooping in the yard again. What's up with people? Just poop in the toilet, you're fine. But if we pooped outside, if we defecated outside, then consumed by this cow who is grazing, that's what he eats, consumes then the parasite. It is going to mature in the parasite, mature in the animal, and again, never ever going to grow up to be an adult until consumed by that primary host. Remember, the primary host needs to have that adult stage. So we have the primary host here, and we've got the intermediate host here. And because this human, it's not like this human just goes out into the pasture and then starts chewing on this cow. No, we usually eat their meat products, or some people choose to eat their meat products. And again, if you eat this particular meat product, medium or below, you run the risk, if that meat is contaminated with this organism, you run the risk of consuming it and acquiring the parasite. Some other terms, habitat, where the parasite lives on or in the animal. So we're talking about what organ or what system the parasite lives. Distribution, we're going to talk about where parasites are found geographically, and ultimately we're going to focus on the ones that commonly infect our environment. Think of like the upper Midwest. We're going to talk about ways that they are transmitted from host to host, because in many times you have the same parasite but has many ways of being transmitted to another animal. We're going to talk about in the first category of parasites, because we're going to break things down categorically. We're first going to talk about the roundworms, then the hookworms, then the whipworms, then the lungworms, then the threadworms, and these are all examples of nematodes or nematodes. And ultimately a nematode or a nematode is a worm that is cylindrical in diameter. So it doesn't get its name, a cylindrical worm. So any sort of type of spaghetti, round in diameter, any of them. Heartworm is another example. We've got lots of nematodes. Then we're going to talk about the cestodes. These are our common tapeworms. These are our flat worms that are segmented. And so if you see this up close and personal, you'll see little ridges. And ultimately, when an animal defecates out a tapeworm, they usually break apart into little segments because that anal sphincter. But a flat segmented worm is known as a tapeworm, belonging to the family of cestodes. We have trematodes, which are commonly known as flukes. And flukes are flatworms. So they're flat, but they're not segmented. And in lab, I describe these more so like as shapy bloodsuckers or leeches. But they're flat, always flat, and they're one piece. We talked in lab, or we'll talk in lab, about protozoans, which are single-celled organisms, one-celled parasites, one-celled organisms. And in lab, you'll introduce this particular one, or know this one, identify this one as Coccidia, and this one specifically is Giardia, more specifically Giardia trophozoite stage. Nonetheless, these guys they only have one cell, and because they're one cell, they're simple animals, simple structures, simple organisms, and because they're simple, they can't reproduce sexually. And so they use a process of asexual reproduction to, again, make more of themselves. Because, again, they're successful because they make a large amount of themselves. Prepatent period. A term that all of these terms we're going to apply throughout the course of the semester. But prepatent period is the time from at the moment the dog gets, or the patient gets infected with a parasite, to the time we can diagnose it on a test. That's very important. Different parasites have different prepatent periods. Throughout the course of the semester, I'm going to introduce you to the prepatent period for all of these parasites. And that can be very helpful, because what if a client just called and said, oh, the neighbor's got a dog, and they just told me that it's infected with roundworms, and I think my dog stepped in his feces and then groomed himself. Should I bring in a stool sample right away? Well, even if there was that transmission, if you tested that stool sample right away, you wouldn't be able to find the parasite, because the prepatent period for many of the roundworms are somewhere between 2 weeks and 4 weeks. So, understanding these prepatent periods from the moment that we get infection into the animal and to the time we can accurately diagnose it. In theory, I'm not going to test you on the specific time frames, like Toxocara canis is 2 weeks. I'm not going to test you on that, but I am testing you on your ability to understand this definition. I am going to test you on one prepatent period, and you will know what that prepatent period is for heartworm, and I'll explain why the importance of that. If you are in vet diseases, you're going to talk about the incubation period. That's something different. That's the time from infection to when you can see symptoms. That's completely different, because many times we're not going to have any symptoms. Just like you, 50% chance of you having roundworms. You're not exhibiting any symptoms. So infection to the detection of parasites using a test. Zoonoses, diseases communicable from animals to humans under natural conditions. So we are going to identify different parasites that are zoonotic. The vast majority of them are not zoonotic. Obviously, we'll talk a little bit more in depth about the ones that are and ways to control or prevent your clients from getting infected, and also you, obviously. Whew, that's wonderful. There was a lot of information today. I thank you for being flexible. This is kind of boring sitting here watching a video. But what I encourage you have access to this video. You can watch it as many times as you want. Oh my God, maybe need a glass of wine. Just kidding. But utilizing this PowerPoint along with your reading assignments, along with your textbook, what other resources that you like, I would like to remind you that in the library they have hundreds of books on parasites. So really cool. I encourage you to watch television shows on the Discovery Channel, Nat Geo. There's wonderful series called, one is Monsters Inside Me. They're talking about predominantly parasites. So a lot of fun tools to help you grasp all of this information. I have provided a worksheet for you, and I'm going to have you tackle this worksheet. This worksheet will review a lot of the terminology that we utilized today, that I introduced you to today. And again, we're going to utilize all of these terms and these concepts throughout the course of the semester. But use the worksheet to review information that we covered today, and that is going to be due next week. Awesome. And I will be back on campus on Monday. So if you have any questions or concerns, please feel free to email me. My email address is on the top of the syllabus, or give me a call, or you can certainly stop by. I'll be in all of next week. Take care. Look forward to meeting you in person.